Hello, this is our Chapter 12 lecture on muscle physiology. So we have three types of muscles that we'll be discussing today. We have skeletal muscles, cardiac muscles, and smooth muscles. Of these three types of muscles, the skeletal and cardiac are striated, and the smooth muscles are not striated. So skeletal muscles have both ends that are attached to a bone through tendons, and when we want to move parts of our body, we're going to have a muscle contract, and we have a bone that moves. It is attached to at the muscle insertion, and there's also a bone that doesn't move, and that's attached to the origin of the muscle. And the movement of muscles is always towards the insertion, and critically, every muscle has tendons firmly attaching them to bone. Otherwise, when the muscle is contracting, the attachment, which is a tendon between the muscle and the bone, could break if the tendon was not strong. Here we see a bone connected to a muscle, and here's the tendon, which is strong connective tissue anchoring the muscle onto the bone. And I want to jump down here and show you that this little unit of muscle is actually a muscle cell, and it has a muscle cell and it has a sarcolemma around it. So there's a sarcolemma surrounding this muscle cell, and in a sarcolemma, by the way, is a plasma membrane of a muscle cell. We also have a unique term for cytoplasm in a muscle cell. This is called sarcoplasma. Okay, that's the sarcoplasma, which is the cytoplasm of a muscle cell, the sarcolemma, which is the plasma membrane. And inside we have these filaments that are composed of cytoskeleton, and we're going to take a closer look at those cytoskeletal factors and how they play a role in muscle contraction. So notably, skeletal muscle cells are multinucleated. So this is one long cell. There's two nuclei we can see that are part of the same cell. And I just want to show you this on this inset. So this is one cell, right? And this is connected in a bundle with other cells, right? So here's this one cell with the sarcolemma around it. Each one of these other muscle cells also has a sarcolemma around it, and they're all bound together by connective tissue. And then as we go up into the more gross level, we see here's one cell. Each of those little red areas represents additional cells, and then these are bundled into bigger groups. Right, here's a bundle, and there's several of these bundles, and each bundle is comprised of many muscle cells. So striated skeletal muscle have these striations are basically indicated by the stripe-like phenomena, and this again is one muscle cell. Look at how many nuclei. There's a lot of nuclei in this one muscle cell. Here's another muscle cell, another one. And what these striations represent are sarcomeres, which are basically units of cytoskeletal filaments that have the ability to contract. And here's this again, one muscle cell, again, one cell. Here's the sarcolemma, which is the plasma membrane, the sarcoplasm. And then in each of these myofilaments, and there's multiple myofilaments in one muscle cell, we have these myofilaments that are really the cytoskeleton of the muscle cells. And again, we see the two nuclei in this one cell. So how do muscle cells contract? So the nervous system, the somatic nervous system that is controlled by conscious effort to move parts of our body, have these motor end plates. So the nerve will have a motor end plate on the muscle, and then it will be secreting neurotransmitter. So this is the presynaptic neuron, and then this is the muscle cell, which is going to receive the neurotransmitter. And here we have mitochondria and then these synaptic vesicles. The neurotransmitter that facilitates muscle contraction is acetylcholine, and the receptor in the muscle cell at these motor end plates are nicotinic acetylcholine receptors. And this is showing a microscope picture of the neuromuscular junction that's depicted in this cartoon. So in order for a motor end plate to trigger a contraction in a muscle, the neurotransmitter acetylcholine needs to be secreted by the terminal of the axon. Then in the postsynaptic portion of which is the muscle cell, right? We have the sarcolemma is surrounding the muscle cell, and we have the nicotinic acetylcholine receptor in the membrane of the muscle cell. So this is going to have acetylcholine bind. It is going to open the channel, and I want to remind you that this channel will have the ability to have both sodium and potassium ions moving with their concentration gradient. So this means that sodium ions will be traveling to the inside of the muscle cell, into the sarcoplasma, which is the muscle term for cytoplasm, and then potassium ions are going to be traveling out. This will lead to this is called the end plate potential. In the nervous system, we call it an EPSP, but in muscle cells, we don't call it an EPSP because this is not a nerve cell, it's just a muscle cell. We call it the end plate potential. So the end plate potential is going to increase the membrane potential inside the muscle cell. This in turn will then go over to number 2 over here, which are voltage-gated sodium channels. The voltage-gated sodium channels will open as the membrane potential increases, and this will lead to a further surge in the increase of the membrane potential. Then next, this will then trigger this number 3, which are these purple receptors over here. This will trigger calcium channels to be opening in the sarcolemma to allow the calcium to enter the cell going with its concentration gradient, because there's more calcium outside the cell in these transverse tubules than there is inside the sarcoplasma, which is the cytoplasm. And what's really unique in muscle cells is that we have, skeletal muscle cells specifically, is that these calcium channels in the sarcolemma are physically attached to calcium channels in the sarcoplasmic reticulum. Sarcoplasmic reticulum is the endoplasmic reticulum, or a smooth endoplasmic reticulum inside of skeletal muscle cells. So because this is covalently, I mean, they're basically physically attached, right, to the proteins, they're attached to each other, the conformational change triggered by the opening of the voltage-gated calcium channels in the sarcolemma can trigger a conformational change in the calcium ion channels in the membrane of the sarcoplasmic reticulum, and then they both open simultaneously. So this is leads to a very rapid influx of calcium into the sarcoplasm. And this is, you'll see in a little bit that calcium is absolutely critical for the contraction of muscles. And I just want to point out that the transverse tubules are traveling through skeletal muscles, allowing for contraction of many units of the muscle simultaneously. And we'll take a look at a bigger picture as we step out of this close-up. So here we see one single cell, right? Because there's one sarcolemma, one sarcoplasma, with these myofibrils, which are the units of the cytoskeleton. And then we also see the transverse tubules, right, that are traveling through out this system. And these play a key role by letting an action potential sent to this particular muscle cell travel through the different regions of the muscle cell. So here's a little summary of what we just discussed. So we have a somatic motoneuron. This will release acetylcholine, and then acetylcholine will bind to the nicotinic acetylcholine receptors, open the ion channels. This allows for both it allows for sodium to enter the sarcoplasma, and it allows for potassium to be exiting. As sodium diffuses in, it'll depolarize the sarcoplasma, and then this will be triggering an action potential. This depolarization will trigger the opening of the voltage-regulated calcium channels that are found in the sarcolemma. And remember, the sarcolemma is going to be the boundary between the sarcoplasma and the transverse tubule. The transverse tubule has more calcium in it than the sarcoplasm. And when the ion channel is open, the calcium will be rushing in to the sarcoplasma. The conformational change in the opening of these sarcoplasma voltage-gated calcium channels will then trigger a conformational change in the calcium channels of the sarcoplasmic reticulum, and then that'll release even higher levels of calcium into the sarcoplasma, and then calcium is going to bind troponin, and we'll learn more about that in a little bit, and this will stimulate contraction. So let's take a closer look at the sarcomeres to understand how the cytoskeletal factors are structured and how they're going to be facilitating contraction. So each sarcomere is a unit of cytoskeletal unit in the skeletal muscle, and each has a boundary of these Z discs. There's a Z disc on one side and on the other side. In between, we see these purple beads represent actin. Okay, purple beads are representing actin. Actin is tethered to the Z disc. And then we have a titin filament, which is another it's an elastic protein attached to the Z disc over here. Another polymer of titin over here attached to the Z disc. And these are covalently attached to myosin in the middle, which is shown in green. And we have some zones that I need to introduce you to. So we have the H zone in the middle, which is a zone of myosin only. Then we have an A band, which is basically where we have both actin and myosin together. Okay, actin and myosin together. And then the region where there's only actin without myosin is called the I band. Okay, so here's the I band, the region with actin alone, and then we have the A band, which is the area with actin and myosin, actin shown in purple, myosin shown in green, and the H band, which is the myosin alone. And if we look in this electron micrograph, we can see that the A band is very dark, right? The H band is very clear, and the I bands are very clear. So the high bands, are going to be very clear. It's the A bands that are very thick when you look under the electron micrograph, because you have all those overlapping cytoskeletal factors.